Hello students, I am NITIN SWAMY, and in this video lecture we are going to talk about Meselson and Stahl experiment. So since we are talking of DNA replication, so in DNA replication one of the prominent question was there in front of scientists was how DNA replicates. The mode of DNA replication was not known in 1958. So in this experiment, in this lecture, we're going to have a broad discussion on Meselson and Stahl experiment. Meselson and Stahl, they were two scientists who proved that what is the mode of DNA replication. Before that, DNA replication is the process by which DNA makes a copy of itself during cell division. In 1953, Francis Crick and James D. Watson, they had written something very peculiar in their paper that was published in Nature that was revealing the structure of DNA. And the last two lines were like, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In 1953 itself, Francis Crick and James D. Watson, they had given some clue about the mechanism of DNA replication, but it was not very clear. Around 1956, there were three different theories which were floating on the floor, and they were conservative model, which was given by Gunter Schint. Second model was dispersive model that was given by Max Tilbrook. And Max Tilbrook was also instrumental in giving that conservative model theory along with Stent. And the third theory was given by Watson and Crick in 1953 itself. Out of these three theories talk about three different mechanisms. The first one was conservative model. It says that one copy would be entirely of DNA that is old and the other one would be totally new. It means parent DNA would always be intact together and new DNA would intact together. Second model was given by Delbruck and it says about dispersive model, and it says that DNA would break into fragments and then those fragments would make complementary DNA strands, and at the end everything would ligate and form DNA. Although dispersive model was not that much accepted because of the non-feasibility of the reaction, and the third model that was given by Watson and Crick says that complementary strands would synthesize and each DNA would be having two elements, one from old DNA and one would be new strand. Although later on, Mises and Stahl proved that semi-conservative mode was a prominent mode of DNA replication. Now, if we see this diagram reveals about the three models which we had just spoken of. The first model is semi-conservative model, which says that parent strand would open up, and after that, in the first generation, after the multiplication, after the replication, one strand would be of parent DNA and the second would be new. Here also and here also. And in subsequent generations also, the scenario would be similar. And after that, in the second mechanism, conservative model, it says that parent DNA would open up first, and after that, they would make their complementary DNA, and again they would split again, and new DNA would come together and form a DNA duplex, and the old DNA would again come together and form a duplex. That this process would go on further in second generation also. Now the third model, that was dispersive model, it says that parent DNA would break into fragments and then those fragments would ligate again and would give a concoction of old and new DNA. In this diagram, can easily make it out that this is made up from new DNA and old DNA, and the process would go further in second generation also. So how anybody would believe that which model is correct, the semi-conservative model, the dispersive model, or the conservative model? So for that, two prominent scientists, that is Matthew Meselson and Franklin Stahl, they had given one experiment that is known as the most beautiful experiment in biology because of its simplicity. These two scientists, what they had done, they had taken E. coli cell. In the normal DNA, we have N14 isotope of our nitrogen in our nucleotides. So what these people had done, they had used a separate isotope of nitrogen that was N15, and they have denoted it as heavy medium. And what they had done, they had kept E. coli cells in heavy medium so that the entire composition of DNA would be N14 always. So in this diagram, when we see, so at sample 0, when they had grown entire bacteria in N15 media, now we can see at 0 minutes, the entire composition of DNA is having N15 isotope of nitrogen in their nucleotides. And they had isolated DNA, and after that they had gone with a process called as density gradient centrifugation. And after centrifugation, they had got one band, that is heavy band, in which is having N15 in both their strands. So they have got one strand, the one band here. Anybody can easily see it here. And now what they had done for the first generation, they had changed the media. Since they had changed the media, so now they are having E. coli cells which would be growing on media which is having N15 isotope of nitrogen. So here we can see after the replication process, there is they are finding one band that is intermediate, that is neither at N15 part nor at N14 part, and it is somewhat in between N15 and N14, and they have called that band like as intermediate band. Now if we see this, we see the interpretation. So the first diagram depicts that the entire DNA composition is having N15 DNA, and the first generation, it is showing that here we are getting two different, here we are getting N15 and N14 bands in DNA duplex. So we can say that parent DNA is also present here and new DNA is also present here, which is giving an intermediate between N14 and N15 band after density gradient centrifugation. And here we are rejecting one theory, that is conservative theory. How we are rejecting it? So since we have already spoken that after first generation in the here, after first generation, we would be having two different DNA duplexes. One would be having old DNA and the other would be having new DNA. So in that scenario, we should have two bands, one at N14 and other at N15, which was not the case. Since they got only one band, so it proved that conservative mode of replication is not there. So they had, after their experiment, after seeing first generation, they had interpreted that conservative model of application is not there. So after that, the second thing now comes, they had to prove whether dispersive model is correct or the semi-conservative model is correct. Because after first generation, both of them would be giving an intermediate band. So because in the dispersive medium also, we had already spoken that it would be having equal composition of new DNA and old DNA. So even after first generation, they would be giving only one band that would be intermediate in between N14 and N14, like semi-conservative mode of application. Like now we'll see after the second generations, when they have grown the same bacteria in N14 media, they saw two bands, one of N14 and the second band would be of N14 and N15. If it would have been a dispersive media, dispersive mode of application, so they should have got only one band, because dispersive mode of application says that after every generation, the composition of new and the old DNA would be equal. In that scenario, we should have got one band here, but we have got two bands, one at N14 and one at intermediate, that is N14 and N15. Now the second generation reveals that dispersive mode of application is also not there. So only one mode of replication which prevailed the entire calculation and the entire observation was semi-conservative mode of replication. And then the scientists proved that the data tells us about that the only mode which shows these interpretation is semi-conservative mode of replication. And in this way, these two scientists proved that something big is there and they had given the mode of replication. And after centrifugation, we can see that there are two bands, one at N14 and N15. And the people they got bands here in intermediate position, which reveals that the mode of replication is semi-conservative. And as we are progressing, and if we go to the third generation, so in third generation, this N14 band would go thicker because with time, the only nitrogen isotope present in the media is N14. So with each replication cycle, this band would go broaden and broaden and broaden, and this band would be there, but this band will always grow, will always increase, which reveals that semi-conservative mode of replication is true. Now, with this thing, I conclude my lecture. This lecture says that although Meselson and Stahl demonstrated that DNA replication replicated semi-conservatively, meaning that each strand in the DNA molecule serve as a template for the synthesis of a new complementary strand, although Meselson and Stahl did their experiments in the bacterium E. coli, today we know that this postulate, what they had given, that theory that they had given, holds true on the all type of organism on this planet. So we should be proud that these two scientists, in this simple manner, had given us something very, very informative, and which was the main basis of DNA replication. Thank you.